Yeah. Okay, for those of you online, we're just waiting another minute or so for a committee member. So.
I gotta be quiet. Yeah, you gotta say something really inflammatory so I can be vehement.
Ah, okay.
Otherwise, I don't know
Okay.
what to be vehement about.
Yep. Kevin, so we already have online comments about Mr. Tassia. Kevin Kosat said he digs his hair. So you all can rate the ponytail. I guess we can do an online survey for the ponytail.
Yeah, I think actually Damien and Corey can make polls.
No,
No, that's the only thing I can do. Yeah. I'll address the online stuff in questions.
At nice ponytail.
Yeah.
Yeah. Hi. Will you run down to Beth's office and see if she's there? You know which office is hers? She's just down the hallway.
No, she's not.
She's downstairs now. Yeah. That's right.
First floor.
Yeah, Paul's just going to text her. Never mind. Stay here, Colin. Thank you, Paul.
Well, she finished. She just got a class.
Oh, that's right. That's right. Okay. Well, while we're waiting for one more person to make their way over, I want to welcome you all to Mike Tassia's defense seminar. Mike is going to be talking about the evolution of Deuterostome immune systems. And Mike came to us from the University of Washington. And so he made that move from the West Coast over to the little village on the plains. One of my first memories well, not one of my first memories. One of my memories of Mike during that time was sitting with him, watching him argue with my then 13-year-old daughter.
She was 11.
11-year-old daughter about who was a better quarterback, Russell Wilson or Cam Newton. Well, after yesterday, I guess we know you won that argument, Mike, because Cam Newton got cut. But so it was really funny.
I've made it.
Finally, you beat an 11-year-old. So like a lot of students at the beginning of their career, Mike came. He was very happy. He had also just finished as an undergraduate. He had helped put together a big review on global diversity of hemichordates, which was sort of the first review on hemichordates in quite a while. So it was a bit of work to put together. And so things were going. And then he comes to Auburn and he follows that up with getting a paper very quickly in PNAS, which was great. It was a very nice paper. He used bioinformatic tools and methods to look for chordate genes in hemichordates, a group that's not studied very much. And it had implications for understanding Deuterostome evolution and Deuterostome immunity. Well, then sort of the honeymoon ends, and Mike really starts figuring out the graduate school is a little bit more challenging. Not everything's a clear-cut answer, and he, some of the questions aren't so easy to figure out. And when he started figuring out there was a lot more information, he sort of became like a deer in the headlights, right? So he realized there was a lot more to learn. And then he thought he was starting to get it. And he's like, okay, maybe I can do this. And then reality started setting in, right? And this is probably the truer face of Mike. This is the true emotion of dissertation research. For those of you who haven't defended yet, you will go through this phase. This is sort of like the epiphany phase where frustration level gets up, gets really high. But Mike handled this well. Mike had his moments of zen. And Mike, knew how to sort of handle the stress. It would get him worked up, but then he'd figure out how to sort of deal with it and figure it out. So one of the things about Mike, which I admire and I think is great, Mike understands how to have a really good balance, right? So Mike has a great, work and life balance, and I think that has helped kept him sane. And I know Kat has helped out with that sanity a lot. But also in his work, he has had a very interesting, for me, gratifying project in that it has balanced organismal work with genomic work, with bioinformatic work. And he sort of has hit on all of those things in his different chapters that you will hear about today. He's done great as far as the dissertation goes. He's got about 9 papers to date. He has 3 already from his PhD. He just got a very nice one into molecular biology and evolution, which is a nice feather in his cap. And he'll have at least 1 more coming out as well. And then Mike's going to be here for a little bit, but he has just accepted a postdoc at Johns Hopkins. So we want to wish him well there, and you all can ask Mike about that later. So Mike's talk. So without further ado, here's the man of the hour who is going to vehemently argue.
Something.
The
Yeah.
only thing I see Mike be vehement about is actually video games. So that's
Yeah. Takes some effort to be a winner. All right. Yeah. Thanks, Ken. Thank you for being here. To people that are on YouTube live right now, I've got a couple people watching the chat in case you have questions. You can ask them, and hopefully we'll address them at the end. But yeah, let's get into it. So the title of my dissertation is The Evolution of Deuterostome Immune Systems, but in large part what I do is I focus on hemichordates and use them as a comparative tool to address the ancestry of immunity in Deuterostome. And we'll get to all those terms in a second. So before I start on the meat of the presentation, I want to give you an outline of what I'm going to talk about. Throughout the talk, you're going to see in the top right corner the part and the slide number. In case you have any questions about a specific part of my talk or a single slide, you can use that as a little bit of a metric so that you can go back, or I can go back and find that. But I'm going to start with some background, talk then about the first data chapter. They'll discuss toll-like receptor pathway evolution in deuterostomes, antiviral differential gene expression in the acorn worm Saccharomyces cowlewskii. Part 4 is this program called Tiamat. And then part 5, I'll talk about some conclusions, wrap things up, and put it into some context. So to start, some background. So before I can get into the background of immunity, I want to get into the background of like why I did biology in the first place. And it all stems from going to the Friday Harbor Marine Labs, where I got introduced to some really wacky invertebrate biodiversity. And though I didn't see like any of these species there, this nonetheless exemplifies the sort of rule breaking that invertebrates kind of do in terms of what we think of biological convention. So like in this cephalopod, the cells that are responsible for causing color changes and textural changes in the epidermis. We have colonial animals that make these beautiful pelagic structures where each clonal individual has its own special function in the context of the entire colony. These are crazy things that don't happen in like the scope of traditional biology as we learn in basic biology textbooks. That fascinated me. What was really interesting that I learned rather quickly was that in terms of biology, we know a lot of the concepts from only a small distribution of species. Humans, mouse, C. elegans, Drosophila, these are the model species that have this long history in biomedical research. But if we look across the animal phylogeny, there's so many diverse body forms and life history strategies that any one person could spend an entire career studying one animal, one body plan. And certainly like that happens. And there's still questions left to be answered. So, in annelids, it's such a diverse group of animals that like you can't see this and be like, that's boring. That's really cool. And even though I didn't get to work on annelids during my PhD, I still got to study some stuff that was rather strange, I like to think. So in the context of animal phylogeny, this bottom left corner image, I focus on this little clade here in the grand scope of things, little, but a lot of diversity in here, and that is Deuterostomia. And Deuterostomia is where we as humans belong. We are gnathostomes. We're jawed vertebrates. Our sister group are the agnathans, the jawless vertebrates, and our closest invertebrate relatives are tunicates. This group is called chordates. We're chordates. And chordates are sister to Ambulacraria, a sister grouping between hemichordates and echinoderms. If you're familiar with either one of these, it's probably echinoderms. They're charismatic and beautiful, and they're in all coastlines of the world. And within echinoderms, we've got sea stars, sea urchins, brittle stars, feather stars, sea cucumbers. They're gorgeous and fun to work with. But I work on the lesser known hemichordates. The hemichordates are sister to echinoderms. Hemichordates or hemichordata is comprised of colonial terebranchs, this little guy on the left, and solitary acorn worms. They're exclusively marine. They're overlooked in both biodiversity surveys and in literature. Very little functional work has been explored within them. And there's very few species that are currently described. However, despite them being overlooked, they do have a lot of importance in terms of providing some phylogenetic contrast and understanding the evolution of what used to be deemed traditional chordate synapomorphies. In particular, pharyngeal gill slits are present in extant acorn worms and extant terebranchs. They're present in some fossil echinoderms, and it was work within hemichordates that helped inform the ancestry of the pharyngeal system that was thought to be a chordate synapomorphy, but instead is now recognized as a deuterostome synapomorphy. Can someone grab the lights, actually? Because I'm going to have some pictures. Thank you. So unlike extant echinoderms, Entrepneusts, the acorn worms on the right, retain a lot of morphological and genetic traits thought to be, thank you very much. Just leave them all off. It's fine. We can get a little intimate in here. So Entrepneusts retain a lot of morphological and genetic features that help inform the ancestry of deuterostome traits. During my PhD, and I'm not going to focus on it here, I did get the opportunity to work on a zoological chapter of hemichordate biodiversity, which was this review of anatomy diversity, taxonomic diversity, and it was a huge undertaking. And I'm not going to dwell too much on it now, but what I want to point out is the major body regions of an acorn worm, because I'm going to refer back to these later. So acorn worms are partitioned into three major body regions. They have an anterior proboscis, an intermediate collar, and a posterior trunk. The proboscis is primarily for burrowing and chemosensory. The collar has, between the junction of the collar and the proboscis is the mouth. So there's some buccal components there. And then the trunk is where the guts, the gonads, and the pharyngeal system are of the organism. Internally, it shouldn't be too surprising. These different body regions also have internal components that are exclusive to the relative or to their section of the animal. So the heart-kidney complex, for example, is in the proboscis. You might infer that this does the same thing in vertebrates, and as far as we know, it probably does. But so moving on from hemichordates, of course, the title of my dissertation involves immunity. Within deuterostomes, there's a lot of cool immunological novelties and divergence that caught my attention very early on in my research. So, of course, we are all familiar with the adaptive immune system, the immunoglobulin-based immune system of vertebrates. This is why we all got vaccines recently, or at least in the last year, hopefully. So this is novel and unique to jawed vertebrates. In jawless vertebrates, they have an analogous system that evolved independently that also affords them a robust response upon secondary infection. And then in echinoderms, there's also a unique immune system component or molecular component to immunity, and that's the SP transformer family. Again, thought to be unique to echinoids and has evolved independently within that lineage. So there's a lot of interesting things that have occurred within deuterostomes, or like novel immunity traits that have occurred within deuterostomes. So what about hemichordates? I didn't put any tick marks in the hemichordate section of that phylogeny. What do we know about them? Well, the last functional work that was done on hemichordates prior to my dissertation was in the 80s, where Rhodes and Ratcliffe injected bacteria into the coelomic cavity of the collar and the trunk, found that those bacteria were phagocytosed by cells. They also did this cursory experiment where they blended up the entire organism and then just like put bacteria in it. And we're like, hey, the bacteria don't live that well. So, for whatever that's worth. And then upon genomic availability, Kate Buckley and her colleagues looked at the evolution and complement of toll-like receptors across echinoids, and because the acorn worm Sachyglossus kalevski's genome was available, included that and found that unlike echinoids, which I'll come back to in a second, Sachyglossus has far fewer toll-like receptors, a component of innate immunity, than in urchins. So given this background of information on immunity work within deuterostomes, given the independent evolution of immunological novelty and divergence within the echinoderms and chordates, I had a couple of big questions. A, what was the immunological platform on which these novelties evolved? And B, how does hemichordate immunity compare? Of course, I wanted to look at a fringe organism and work on something weird, so hemichordates were the subject for me. So now I'm going to jump into the first data chapter, toll-like receptor pathway evolution in deuterostomes. So before we can get into what I did, I want to give a basic idea of how immunity works. So there are immune cells of many different types. A lot of these immune cells express proteins on their cell membranes that can bind conserved molecular patterns that are indicative of pathogens. So gram-positive bacteria have peptide glycan on their cell walls, double-stranded RNA of viruses, lipopolysaccharide of gram-negative bacteria. These cells and proteins are able to recognize those patterns. They can then phagocytose, in some cases, phagocytose the bacteria, digest and dispose of the waste, and then they signal via a signal transduction network that starts at that receptor to express pro-inflammatory cytokines, antibacterial peptides, antiviral peptides, so on and so forth. So immunity in general is a combination, at least as we know in vertebrates, a coordination between the native immune system and a learned immune system, i.e. the innate and adaptive immune systems. Innate immune systems don't change over the course of an individual's lifetime. It's all genome encoded and relatively static. And then the adaptive immune system leverages molecular learning, for lack of a better term, or immunological memory, to respond more robustly upon secondary infection of a pathogen that's already been present within the system. And then it is also a delicate dance, of course, between cellular and molecular components. So I mentioned briefly the toll-like receptor pathway earlier. That's the subject of this chapter. The toll-like receptor pathway is an innate immunity pathway. It doesn't change over the course of an individual's lifetime, and it is molecular. So the toll-like receptor pathway relies on transmembrane proteins that are either localized to the cell membrane or the endosomal membranes. And upon ligand binding, they go through a signal transduction network that activates activates nuclear factor kappa betas and interferon regulatory factors that regulate the expression of those molecules I mentioned earlier, pro-inflammatory cytokines, antibacterial peptides, et cetera. And this pathway on the left, which all kind of is initiated through these domain-domain interactions between the toll-like receptor and a domain on the toll-like receptor called the TIR domain and the TIR domain of another protein called MID88. This pathway on the left is thought to be the canonical pathway. There are homologs in Drosophila, but there are alternate adapter molecules that also leverage homotypic domain-domain interactions to modulate how that downstream transcriptional effect will change. And importantly, the number of unique toll-like receptors that are encoded, toll-like receptor protein species encoded within a genome has implications for the breadth of pathogens that can be recognized for the individual. So where one organism has 10 toll-like receptors, you might expect that it can recognize fewer pathogens than an individual or a species that has 5 million. That's totally hypothetical. So what do we know about toll-like receptor diversity within deuterostomes? Starting with vertebrates, we have 10. As humans, mouse I think has 13, but it's on the order of about that scale. And genomic availability provided a rather unprecedented ability in the 2000s, 2010s to look at the number of toll-like receptors encoded within a genome. And starting with tunicates, they had fewer than 5, which is of course a reduction of less than the roughly 10 recognized in jawed vertebrates. Urchin has over 200, which is a huge expansion and really interesting. Over 70 in the lancelet, and then in Sachyglossus kalilevskii in the paper that was focusing on echinoids, the genome of Sachyglossus kalilevskii only possessed 7. So this led to the paper Toll-like receptor pathway evolution in deuterostomes, where there were a couple major aims. So the first aim was to investigate the full toll-like receptor pathway, not just the receptors themselves, using a unified bioinformatic protocol allowing us to make apples to apples comparisons between species. As you may know, if you do a lot of bioinformatics, differences between computational protocols can make a little bit of cause a little ambiguity when you're comparing data sets. And then I also wanted to improve the representation of hemichordates in this context because they had only been looked at relative in very few numbers, and we had a huge phylogenomic data set of hemichordates that we could leverage for this study. All right, so how do we do this? So starting with transcriptomes and genomes across 39 different taxa, 49 total data sets. We had replicates for some of them. We started with transcriptomes and coding sequences from genomes. Transcriptomes were only looked at for presence. Genomes were looked at for presence and absence of components. And these were translated into peptide sequences. They were clustered so that we were only looking at individual unique proteins per genome or transcriptome. These were then assigned putative homology using the Swiss prot protein database. So whatever the best hit target was, if it was something we were interested in, it was part of the canonical toll-like receptor signaling pathway, we retained it. Anything that was putatively homologous was then annotated with protein domains, which gives us this modular perspective on whether or not the protein is built as it ought to be, if it is homologous and functions the same way. Then these proteins were placed into a phylogenetic framework to get to validate homology and try and distinguish orthology groups among the proteins. So importantly, this method looks at the unique number of proteins per species, not what's within the genome, if there's synonymous genes, for instance. And it considers only protein homologs that could be validated by domain architecture. And this is important for functional inference. If we're looking for a toll-like receptor, it needs to be built like one. If we're looking for the adapter proteins, it needs to be built like the conventional adapter proteins. All right, so just a reminder, toll-like receptors interface directly with the pathogen-associated molecular patterns, and toll-like receptor orthologs are ligand-specific. So, for instance, one toll-like receptor might bind double-stranded RNA, and another might bind peptidic glycan or flagellin. So first, we found that this canonical pathway, which was familiar in other invertebrates, was conserved across all the Deuterostome taxa that we looked at. This is the mid-88 dependent pathway. This indicates that the last common ancestor of Deuterostomes likely possessed this pathway on the left. Whether or not it was functioning in the exact way, is uncertain, but it at least points at this is a common immunity toolkit that was inherited by the crown groups within Deuterostomes. Using phylogenetic methods, this is a Bayesian phylogeny of toll-like receptors. All nodes on this have at least 95% posterior probability. I could talk about any one of these nodes and clades in and of themselves, but I'm going to focus in on one in particular that drew our attention, and that was a clade which contained human and mouse toll-like receptor 3 within a distribution of invertebrate toll-like receptors. And toll-like receptor 3 is really interesting because it recognizes viral signatures. It binds double-stranded RNA. And toll-like receptor 3 in vertebrates, where the functional work has been done, signals via TRIF. TRIF is one of the 5 toll-like receptor signaling adapters. It is not mid-88, which is on the left. But unlike mid-88 and some of the other proteins over here, TRIF was absent across the invertebrate taxa that we looked at. We couldn't find with any confidence TRIF homolog that was built like TRIF ought to be. So this was really interesting. So given the conservation of toll-like receptor signaling components, toll-like receptor 3 orthology, and lack of toll-like receptor 3's conventional signaling partner TRIF, this led to some rather interesting questions that kind of led into the rest of my PhD. First, does Saccharomyces cowlevskii toll-like receptor 3 ortholog display functional conservation with its mammalian counterpart? If toll-like receptor 3 in Saccharomyces works like toll-like receptor 3 in mammals, is it endosomally localized? Does it bind double-stranded RNA? Does it activate interferon regulatory factors? Next, more broadly, can hemichordates even recognize and respond to viral infection? This is just kind of an expansion of the toll-like receptor 3 avenue of thought. And then finally, do other innate immunity pathways display a similar level of evolutionary conservation across deuterostomes as what we found in toll-like receptors? And this was all part of an NSF grant that Ken, myself, Beth Schwartz and Rita Grays were awarded. So we actually got to address a lot of these questions and are continuing to address a lot of these questions. So that was really exciting for my PhD. And in particular, I want to point out that this first one, the functional conservation of toll-like receptor 3, the way that we're addressing this is rescuing toll-like receptor 3 dependent signaling phenotypes in human cell lines, rescue of a kappa beta or an interferon regulatory factor driven reporter. This is still ongoing. Huge shout out to Haley Hallowell, who is doing a lot of the bench work and is truly doing the lion's share of this work. I would be happy to talk about it. I'm sure she would too. But I'm going to skip forward to the next part of the data where I have some pretty cool results to share with you. All right, so antiviral differential gene expression in Saccharomyces. So this addresses the question, can hemichordates, specifically this species on the right, Saccharomyces cowlevskii, can it recognize and respond to viral stimulus? The vague idea here is let's just poke them, let's make them sick and see what happens. And the very specific science fancy way of talking about it is I want to virally stimulate differential gene expression, or at least that was what I was hoping would happen. So to do this, we used high molecular weight and low molecular weight poly-IC. This is synthetic double-strand RNA analog known to be a potent agonist of antiviral immunity in mouse, among other species. And these were injected into individuals in the proboscis, and the gene expression of these individuals was compared against individuals that were injected with just the buffer itself. And what was fun about this and kind of useful for making the methods work was that we coupled this with a fluorescent marker, allowing us to look under fluorescent microscopy and identify the territories in which the injection solvent was within the animal. It's a little difficult to see, but it basically, if we inject it into the posterior portions of the proboscis, it diffuses within the proboscis. And then in some cases, we want to get these back on ice and put them back in their normal setting as quick as possible. But in some cases, in practice injections, we could see this transmit to the other body regions. So starting with the whole individual, we are comparing the ones that are injected with poly-IC to the ones that are injected with just the injection buffer. This was done in a time-dependent way where we could look at the expression at 0 hours post-injection, 2 hours, 4 hours, 8, 12, 24. These were compared against one another. They were done at the same time. All individuals were female. They had all spawned within 72 hours prior, at least 72 hours prior to the experiment, to try and get away from any genetic variants that might be derived of things that we're not really interested in. And this was done in 4 experimental replicates over a couple of years at MBL. After these time points, individuals at their sacrifice time were trisected into their individual body regions. And this was important because we wanted to get capture the systemic effects of antiviral gene expression. So if we look here at this PCA of dispersion between the libraries themselves, we have clustering by proboscis, or trunk, proboscis, and collar. So by controlling for this explicitly in our design model in the statistics, we're able to then capture the more nuanced characteristics of differential gene expression by time and condition. So this, again, is not that surprising. Like I mentioned earlier, each of these body regions is highly specialized, so we needed to control for that. This also means that in post, we can go back and look at or region-specific transcriptional differences. So these libraries, 48 individuals, 144 libraries, 48 times 4, or 3 for the 3 body regions. We looked at the log fold change. In particular, we wanted to know the change in expression due to treatment versus control, which is the reference condition, and the added effect at each time point on the conditional expression of a gene. And we included time, post-injection, and tissue as random effects to basically capture the variation due to those. Weren't interested in them, but they allowed us to discern the genes that were significantly changed due to viral presence or double-stranded RNA presence. And first and foremost, the coolest thing about this is that we did find many genes that were regulated upon viral or double-stranded RNA presence. So this wasn't known prior to this work. This is huge. The fact that hemichordates can recognize and respond transcriptionally to viral presence is a very critical point for understanding the evolution of immunity, particularly antiviral immunity in deuterostomes. So a little bit about this graph. The y-axis is the number of differentially expressed genes that were above our significance threshold or within our significance threshold. they had walled p-values below 0.05, which was our FDR, our false discovery rate threshold. And then the x-axis is the time post-injection, where each of these time points is a categorical variable. So this is 2 versus 0 hours, or expression change at 2 versus 0, 4 versus 0, 8 versus 0, 12 versus 0, 24 versus 0. So at this first time point, we see that, and the red line is down-regulated genes, green is up, and blue is the absolute value, or the absolute number. So this first time point, 2 hours post-injection, we saw an early down-regulation of cellular and metabolic processes. This is consistent with what you might think happens when viruses are being dealt with. Cells are, not replicating anymore if they were in the first place. 4 hours post-injection, we see a huge upregulation of inflammatory response genes and apoptosis genes, as well as many genes that could not be annotated using the method that we use, and I'll talk about that in a second. And then finally, we have this late refractory period where the patterns weren't that clear, but there's clearly some amount of expressional change between these time points. And only one day is as far as we kept the animals to. The picture of this, is our first glance at how antiviral immunity works. It would be really interesting to go this way. It would be really interesting to expand these gaps and look at the finer points. But this is, huge step forward in and of itself. So coming back to annotation, the way that we annotated these genes that were differentially expressed was, again, leveraging a similar method that we used in the toll-like receptor paper, taking differentially expressed genes, assigning them their best hit ID via the SwissProt database, a curated database of proteins with function, ideally. And then these were then pumped into Panther, which then gives a little bit more broader functional context per gene. And this is all a similarity-based method. So for instance, at the time point 2 hours post-injection, among those genes that were downregulated, which was the largest pool of genes, we had many pathways that were downregulated that you might expect, such as like angiogenesis, some immunity stuff like interferon gamma signaling, inflammation. Those were downregulated and then subsequently upregulated 4 hours post-injection. And this isn't surprising either. It might be counterintuitive, but immunity works in this very closed or tight circuit where you don't want to overexpress or underexpress any of the immune response genes because you don't want to cause autoimmunity due to overexpression or destroy host tissues, and underexpression might yield inefficient immune response, and then you're just left with still being sick. I mentioned in the prior slide that there were many genes at 4 hours post-injection that we couldn't annotate, and that's actually true across the board. And that's actually, I think, maybe my favorite part of this is that approximately 40% of the genes at each of these time points that I just threw the pie charts up with, we couldn't annotate at all using similarity methods. So they don't have anything within the SwissProt database that is similar above the threshold that BLAST uses, which is rather loose in and of itself. And these may represent any one of several things. It could indicate that hemichordates have novel immune factors. It could indicate that ambulocrarians, that is hemichordates and echinoderms, possess a shared immune system that isn't recognized in echinoderms, and that's actually may be true given some antiviral work in echinoderms. Of course, it could also mean that this is an ancient antiviral immune response complement that has just been lost in vertebrates. But a lot more phylogenetic methods and deeper taxon sampling within deuterostomes is necessary before we can come to any of those conclusions. Okay, so final data chapter that I'm going to talk about today is about a program I wrote called TMAT, leveraging the title of the paper, Leveraging Biodiversity to Revise Protein Domain Models. So like I said in that slide where I talked about the subsequent questions after the toll-like receptor paper, this was kind of just one of many innate immunity signaling pathways that we were interested in. There are many others that aren't going to be included in this very noisy diagram, which I recognize is up here now. So toll-like receptors, Rig-like receptors, Nod-like receptors, these all work in very similar ways where they bind pathogen-associated molecular patterns. They help cells recognize pathogens that are infecting a tissue or cell. And then they all converge to some degree on NF-kappa B or interferon regulatory factor activation. So we wanted to look at these pathways in an evolutionary context. Before I get into that, coming back to how we characterize uncharacterized proteins, or at least try to, without functional work. So if we have this hypothetical sequence, Sagaglossus seq1, I suppose is what I called it. In retrospect, I should have named it something quirky. This protein we can assign some similarity-based ID to using the Swiss Prot database or NCBI or EMBL. There are many databases where proteins or genes have IDs that are derived or assume some amount of homology to proteins that are of known function in model systems. So this sequence within the Swiss Prot database or the UniProt knowledge base is most similar to mid-88 in human. So this is somewhat informative, but it's only a weak inferential metric for function because we don't know how it's built. We don't know about any of the relationships between this protein hypothetically and the vertebrate proteins. And one way that these similarity assignments of ID are validated is through domain annotation. So if we know what it is similar to, if it works the same way, maybe it's built the same way. So I talked about protein domain annotation earlier. I'm going to dive much deeper than anyone probably wants to hear into it. So if we take a protein and annotate it using hidden Markov models in the PFAM database, which is this huge database of well-curated protein motifs, we can get an idea of how the protein is structured in its sequence characteristics. So for instance, mid-88 is this adapter protein that is responsible for transmitting an activation signal from the toll-like receptors to the other toll-like receptor signaling network proteins through tier-tier interactions. And canonically, tier-tier interactions occur between a single tier domain in mid-88 and a tier domain of toll-like receptors. And then the death domain on the other side of mid-88 interacts with another death domain. When there are other protein domains incorporated into the protein structure as a whole, this draws a little bit of ambiguity as to what other things it could be doing, given that conventionally we think mid-88 is just a single death domain and a single tier domain. You might think that if this has a hypoxia factor, that would also be very strange. So let me talk to you about the PFAM database. So the PFAM database was established in the late 90s, and all PFAM-A database entries are built the same way. So I'm going to talk about the tier domain for the most part, but there are other proteins that we considered, or protein domains that we considered in the study. So starting with a seed sequence, the PFAM database aligns all of these seed sequences from proteins that are known to be related. And this is the related motifs for the tier domain. So here we have toll-like receptor 4 from human, interleukin-related L1, whatever it is, I don't remember right now. But these are all part of the same tier domain containing family of proteins. And the residues are aligned using alignment software that includes gaps and includes variants so that we can construct models, which are represented here as a logo, that capture the variation per site of homologous sites within the domain itself. So this logo, which is kind of the easiest way to look at a lot of data, I'm going to walk you through this. The absolute y-axis here is the relative information content of that site. The relative height of a single amino acid character relative to the other amino acid characters are the emission probabilities of that amino acid per site. You can draw a line to the probability that this amino acid occupies that site in the empirical data set. The other thing that these Markov models capture is the probability that it's occupied at all, which for the most part is usually 99% because that's what it defaults to. And then the probability that there's an insertion following the site and the length of insertion that may follow that site. So in this case, some of these sites are information rich, some of them are information poor. It varies from domain to domain, and they are independent between domains. So the tier domain construction is independent from the death domain construction, for instance. So this allows you to capture, instead of just relying on similarity metrics, actually empirically measure the variance per site of homologous residues within a domain, which is useful when you're working on non-model systems. So in the PFAM database, this is then folded back into PFAM-seq, this proteomic database that provides evolutionary context to the distribution of species which possess a tier domain. So looking at PFAM as a whole, if we plot the number of occurrences that a PFAM domain can annotate a protein within a species, we have this distribution of species and occurrences where human is really well annotated by PFAM domains. That's not surprising. There's been a lot of human work. There's a lot of human genomes. There's a lot of human-based models. mouse, Danio, Branchiosma, Lingula. Lingula and Branchiosma are worked on far less than these organisms, but that kind of indicates the power of capturing variants and modeling a protein structure rather than relying on similarity alone. However, when we look at the number of sequences that are actually within seeds themselves, the sequences that are being used to build the models, so that's in blue here, the species that are most commonly incorporated, so the top 5, are human, mouse, Danio, Drosophila, and C. elegans, the classic biomedical models that are used for many different studies. So this may indicate, in the hypothesis that we had, that the models themselves are only capturing biodiversity of a few discrete taxa and thus don't appropriately capture variation of homologs that are maybe in Apostacus or octopus. So to address this question and frame it in the context of immunity evolution, I developed this program, Tiamat, with the help of Kyle, my friend James, Ken. And the aims of this program was to revise the protein domain profile and Markov models to capture homologous sequence variation from underrepresented species. And I did this, of course, and applied it to innate immunity proteins across metazoa. So no longer is this study focusing just on deuterostomes, but it does heavily sample within them, but looks across metazoa animals. So as a case study, the example I'm going to have up on the screen is for the tear domain. Again, that domain that's central to the toll-like receptors and interleukin 18 receptor family. And we looked at 39 metazoans across the animal phylogeny. And the basic idea of how this works, and I can go into the nitty-gritty if you'd like, it starts by looking for proteins that are the best hit, given the hidden Markov model, to the domain you're interested in. So it goes through every single proteome, finds the proteins which may have a tier domain, annotates it with every possible domain, and if a tier domain is still the best hit of all other domains within the database, it extracts that sequence from the parent protein, aligns it to the model and the seed, and then readjusts the properties of the hidden Markov model and searches again. So where the tier domain may appear in a logo fashion like this before domain revision, after domain revision, it looks like this, and it may look very similar, and that's because it is. The asterisks are the sites where the consensus sequence, the most probably emitted amino acid character, has changed. So the leucine has switched to a tyrosine, the isoleucine to a leucine. I may be getting these amino acid characters wrong, but the letters don't lie. But what's really interesting is that across all the domains that we revised, the effects were consistent across the revision process, where the consensus structure remains relatively conserved. It's these high information content sites usually don't change. Actually, never changed in any of our case study domains. And the adjustments primarily change as information content per site, the relative emission probabilities. So for instance, this arginine and isoleucine is now R and K, which I won't try and pretend I remember the amino acids for. And this shouldn't be surprising that it stays consistent, because the first step of the entire process is only to incorporate domains that are found to be the best fit for the domain in the first place. It's constrained to always incorporate only the domains that are hypothetically homologous according to Hammer's homology definition. So going back to the tier domain and toll-like receptors in general, again, toll-like receptors are structured with an ectodomain of leucine-rich repeats. This convokes the ligand binding properties, and then a tier domain, which is how the signal is transduced through the signaling pathway. And the model before looks like this. And in the toll-like receptor paper, we saw this, and of course now we saw it here with Tiamat's paper, where there are many proteins that have leucine-rich repeats. They are best hit to toll-like receptors, but they lack the tier domain to confidently be inferred to act in the pathway. Leucine-rich repeats are not exclusive to toll-like receptors or any related pathway. There are many different proteins that leverage leucine-rich repeats. So in the toll-like receptor paper, all these would have been thrown out. After domain revision and searching again using the tier domain, all of these proteins had tier domains in territories where we might expect them to be if they are homologous to some degree, or if they are homologous to toll-like receptors. Now, they may not function the same way. We had to capture or revise the domain in order to find it, but they are homologous, though may be more distantly related and may not function in the same way as other toll-like receptors that were identified before. So we, again, not just looked at toll-like receptors for this paper, we looked at many different pathways and the interferon regulatory factor family and NF-kappa B family, built domain diagrams for every protein. And by built, I mean I wrote a program, which was really fun to write, that will just generate these for you, and built phylogenetic trees of all the proteins that we were interested in: toll-like receptors, Rig-like receptors, Nod-like receptors, interferon regulatory factors, and NF-kappa B. And I'm just going to skip to the big points and finding of this. Toll-like receptors, Rig-like receptors, and Nod-like receptors are affected by database bias. We found novel members of all these families after domain revision, which is really interesting. And there was a lot of protein diversity that was not necessarily recognized beforehand. And in a broader sense, not just Deuterostome-centric but viewing innate immunity through the lens of model species appears to misrepresent its evolution across Metazoa, in particular, the diversity of proteins that may be acting in innate immunity pathways. All right, conclusions, closing statements. During my PhD, I think that there's been a lot of cool stuff that we've been able to find on on innate immunity evolution, leveraging primarily these organisms, but also like big genomic and transcriptomic data sets. The toll-like receptor pathway paper was able to confidently find that the MYD88 dependent toll-like receptor pathway was likely present in the last common ancestor of deuterostomes. Toll-like receptor 3 is the only toll-like receptor where orthology could be supported across deuterostomia. This has really interesting implications for antiviral immunity. Saccharomyces does possess an immune system that is capable of recognizing and responding to double-stranded RNA injections. And the differentially expressed genes include both genes that can be annotated for function via similarity methods, but also have a lot of genes that appear to be totally uncharacterizable by similarity methods. Finally, Tiamat taxonomic bias does appear to affect PFAM database annotation for non-model species in particular, and innate immunity protein family diversity appears to be misrepresented due to a deep focus within biomedical models. And something I didn't talk about with the Tiamat. Tiamat is constructed in a way that allows you to use any species you're interested in, as long as you have proteomic data sets, or just a collection of proteins, not like proteomic sequencing, and any domain within the PFAM database. So if you want to appropriate Tiamat to look for in the paper we talk about, looking at oxygen binding proteins within vent species on the bottom of the ocean, you can totally do that with that. So what's next for immunity evolution? I think I made more questions than I answered with hemichordate immunity, which is the sign of a good system, I think. There's still many questions left to answer. It's been enjoyable to get to where we are now. Immunity is even more anomalous in the earliest diverging lineages of animals. I love ctenophores. I would love to do basically the same experiments in ctenophores. I don't think it would be that hard. Famous last words. And something I didn't touch on in the Tiamat paper was there appears to be a lot of domain substitutions within the death domain family. Seemingly, there's the domain is rather promiscuous between proteins involved in apoptosis and immunity, meaning that there might be some evolutionary connection between those disparate pathways in the terms of immune responses. And this is just the beginning of what might, I don't know what's going on there, but it is a pattern that seems to be true. All right, following all of this work, there are some things that are in the pipe for following my PhD. I have developmental time points for saccharomyces, and I've done basically the same experiment in juveniles, where we can look at whether or not the hatched juvenile has an immune system. Does it respond the same way? And I also have all these developmental time points to try and look at the initiation of immune response or like immunity genes when they turn on in development. And then we also have genomes. This is my single shout out to Terabranks. We do have a really awesome genome that's coming through right now for Cephalodiscus hodgsoni, and hopefully for these other 3 species that we collected in the Antarctic this last December. So that's something that I have on my horizon as well. Over the course of my PhD, I've had the luck to work with some really awesome people on some really weird projects. And I say weird in a very positive way. It's basically the reason I do science in general. And I want to give a shout out to some really cool papers that I got to work on. And I could talk about these ad nauseum. And I want to thank people throughout my PhD that have kept me sane. You guys are awesome. And my family and these lovely cats that, keep me sane to this day. And thank you. Okay. I'll take any questions. If you have a question in the chat, you can message, and I think Damien will keep an eye on that for me. I can turn the lights back on, I think. I got it. Cool.
I said you could go the first time.
Oh, okay, great. Paul.
Yeah.
Thank you. I didn't actually write down the slide number, part
It's okay.
3, slide 5, something like that. You showed the genes that were affected, but one of the groupings that you said were affected was B cell activation.
Yeah.
What's up with that?
I
So is that misadaptation?
No, so like there are genes that are involved in B cell activation that, like, for example, like TGF beta genes are known to be present across like animals. And I don't know if this is true for B cell activation, but they certainly could be involved in the activation of B cell responses. And it's likely that the gene that was annotated with B cell activation is just part of some signaling pathway or transcriptional response for differentiation.
Okay, so they're not, but in that analysis, it should be, it wasn't like network, you know what I mean? Like you had to have multiple parts of B cell activation, or just one gene by itself that's left over? I mean, they don't have adaptive immunity, right? So they don't have B cells.
Not that we know of. It's certainly not homologous.
Not that we know.
And this here?
Yeah.
Yeah, B cell activation.
Correct.
Yeah, it could just be a differentiation factor. It could just be some gene that initiates differentiation. And in vertebrates, where this is all mostly based off of, it promotes differentiation into B cells. Whereas for other things, it could just be like some immunocyte, some phagocytic cell type.
But you did it on whole animals, right? So you don't know if they're expressed in melanocytes?
Yeah, this is whole animals. Well, tissues, but yeah.
Right.
Yeah, we didn't do any cell isolation stuff. I actually tried to, instead of pushing fluids into the cavities, I tried to pull fluids out of the cavities.
Yeah.
And it was such a pain just to get like the needle to stay within the cavity, let alone like do anything else. My first trials, I actually injected them too much, which obviously weren't included in the data set. And
You can't just cut them. That's what they do for things with arms. Just cut the arm and sort of squish them out.
possibly.
Yeah.
Yeah, certainly could try that. I tried to see if I could inject a fluorescent marker into the circulatory system as well. Didn't work. Way too hard.
Are any of your toll-like receptors developmentally regulated?
There's no way of knowing for right now, unless looking at, oh, you mean in the differential expression stuff?
Or no, in your time course. Didn't you do an embryonic?
Oh, we don't have any of that data yet. It's all sitting in a -80. Maybe. It'd be cool to know. Any questions online? Hi, everyone. Nope. Cool. My committee, I'm sure you'll ask me
Another
questions.
question, yeah. Then you'll have plenty of questions.
I got a question about the using TMAT.
Yeah.
And when you remodeled the domain,
Uh-huh.
looking at, I have obviously no idea about NP computational, but the ability to then say in that organism corresponding changes in things that it's meant to interact with. So do you find that there's cryptic TIR domain and then there's cryptic YD88 domain? Are they both changing at the same time?
So mid-88 has tier domains. Are you asking, like, are there many proteins where we see, like, they are not related to toll-like receptors, but then they just have the domain all of a sudden?
Like I guess maybe I don't know enough about this. Like yes, you have like a toll, ones that you said weren't a toll-like receptor, and then you say yes, now it does have that domain.
Yep.
does it have the partners or not? Which is where you started, right? Like you wanted to have the toll-like receptor and the partner. And I'm just wondering
Right.
whether the partner's drifting at the same rate.
That's a really great question. Didn't explicitly look for that, but because the tier domain within the adapter proteins and the toll-like receptors themselves are the same domain as far as like the domain model, if we revise the domain for the toll-like receptor, so those 5 that I threw up here, those are all 5 that we found across all of metazoa where they now look like toll-like receptors and have that domain. There is a lot of diversity here. So there's a lot of diversity. So these are from Marco Gerdau's paper, evolutionarily conserved tier domain containing protein families, where there's just like wildly weird protein domain architecture that have tier domains. And this could just mean that the tier domain isn't, only part of the toll-like receptor pathway and a related innate immunity pathway, but part of other things. So if we revise the tier domain with a proteome, we find all of this other stuff. The program itself is domain-centric, not protein-centric. So it doesn't care where it finds it as long as it's homologous, which was an important part of the program.
But your domain architectures are predicated on gene models, which are predicted somehow,
Yeah.
I assume. So how many of these peculiar domain architectures are supported by transcriptome data or something more real?
Yeah, totally. And that's absolutely a good point. All of these papers were from a transcriptomic, or all these domain architectures were described from Lophotrichozoans, Lingula, Chrysostria, so an oyster, where they use transcriptomics and genomics. And there were some subsequent papers by this group that does machine learning on protein family clustering. They also found these in some mammalian systems, in some early diverging teleos lineages. So it seems like these are, to some degree, real. I think maybe we just need to loosen our definition of tier domains, or like within PFAM, there's also like 4 tier domains that don't really make sense why they're partitioned from one another in the first place. But yeah, gene modeling, it is a circular argument if every genome is based off of, or genomic analysis is based on gene models.
And maybe you should comment a little bit about using the molecular data to look versus what you can actually do functionally.
Yeah,
And
so if you wanted to test, like ultimately, if you wanted to see whether or not these tier domains are the same as like if this was from human, you would need to do some like protein-protein binding assays to see whether or not that motif does still bind with other tier domains, if it has the same loops. And that can't confidently be inferred using protein structure inference. Although there is that new machine learning program that's like made a huge splash. Maybe that's something to look at.
There's a reason that's worried.
Yeah, Kyle, can you on that? Thanks.
So 2 questions. So is this system amenable to CRISPR? Would you be able to go into these novel potential PRRs?
So what system? Like saccharglossus, hemichordates?
Yeah.
So there is a group working on establishing CRISPR in saccharglossus. It's thus far been unsuccessful. As far as this is like 2-year-old information because we had that whole pandemic thing and I fell into a hole and disappeared for a while. So unknown, don't know. I don't know if CRISPR, I think it would be much easier to get the gene into an expression vector, isolate the gene or the translated product, and then, or at least take a subsection of it and then just purify that protein and do some protein assays. I don't know how to do those, but I do know they're certainly possible to do. But that's probably the first avenue I would go about trying to test the function of the domain, or at least the biochemical properties of it.
So, and you said about 40% of the genes that you pulled out, couldn't make any sense out of them. How would you? Seems to me they could be kind of important.
Yeah, I also I think those
how
are
would you
really
go about attacking that?
Yeah. So these genes, this is just talking about how we looked at the broader functional annotation. And this is Panther pathway data. There's also other Panther annotation methods that rely on the same diamond BLAST IDs. There's Go term annotation. And what this category, this 60% is all the genes that couldn't even be assigned Swiss prod IDs. What we did do with them is annotated them with PFAM domains. So we might be able to plug those into, there's a program that like infers protein-protein interaction partners due to domain architecture. So we might be able to plug those in and see if any of them are like correlated with one another in terms of interactions. But most likely it's just going to remain as a domain annotation thing, and then we can like count the number of domains spread across the species. It's going to be hard to address those when they have no homologs in the node systems. Yeah. It's interesting. I think it's really cool. Yeah.
Yeah, and just sort of to reiterate on those charts, all those annotations, those are what the homologs do in model organisms, right? So that is what that gene does in mouse.
This is also maybe a little misleading because this doesn't mean that one, or let's say this is 10%, maybe, I don't know how to count pies. This doesn't mean that 10% of the genes are involved in inflammation. It could mean that 2 genes are involved in inflammation and then everything else is 1. One gene might be associated with like 5 of these terms, and another gene might just be associated with 1. So this is to some degree misleading, but it is, with careful dialogue, what we have to rely on to make some functional inference in the system.
Okay, any other questions? Nothing from the chat table.
Hello, congratulations.
Well, that's a little premature.
Yeah. No thumbs down, so only thumbs up.
No, only thumbs up, yeah.
Let's see if I can see them. Hey, everyone.
You're gonna see that.
Okay, well, Michael, thank you.
Yeah.
Appreciate it, and thank you all for coming. Once everybody exits, we can figure whether we want to stay up here or not. Good to see you, Tony. Probably next week. I expect they're October 1st, but I close on my house next week. So I decided to go around the house with Alpine for 2 weeks. Working there. And apparently the house is still there.
Yeah, right. What's that?
Oh, maybe, I don't know.
Stay in touch.
Yep, I will. Yeah, so.
This is the ASMR part of the talk. Thank you for joining. I've got snacks downstairs. I can bring them up here, but it would be easier to be in a closed room.
Yeah.
Okay, we can do that, or we can ask somebody to bring them up.
Either one. I don't care. Either way.
Okay, go to that. Is that okay, Mike?
Yeah, totally. I could like maybe stretch my legs and go down and get them if that's okay.
That's good. Yeah.